Hi, hello, and welcome again. Microbehunter here. Here we are. Here we are again. Welcome to another Saturday microscopy live stream. There is some echo here, which I'm going to quickly turn off. Okay, here we go again. Yes, it does feel a little bit like Christmas again today. Look what I brought along here. Yeah, I need to explain this a little bit of why I've got so many microscopy accessories today. I would like to show them to you. And later on in the video, of course, I also want to do a little bit of, again, like every week, a little bit of practical microscopy as well. I've got a whole bunch of, I don't know, 10 water samples over there, some of them pretty old that I would like to look at again. I did try them out, and there are a couple of microorganisms in there as well. Yes, a lot of people started to join in again. Usually during the first 3 minutes or so, I just say hello all around the world. This gives the viewers the time to join in into the live stream. Yeah, so hello from Vietnam, from Hungary, from Athens in Greece, from Germany, Belgium, Northern France, the UK, United States again, from Texas and North Carolina. Okay, there is a question already. I'm going to try to answer, underline the try, to answer the off-topic questions later on. From the Netherlands, Morocco. Yes, Colombia, Philippines. Yeah. How are the slime molds doing? I'm going to show them to you. They're doing fine. Yes, they've grown quite a bit. From the UK, from Finland, from Montreal, from India. Yeah, that's really nice. United States again, Connecticut, from Sweden. Okay, and from Yorkshire, and yes, very nice. Yes, here we are again. Okay, so I need to explain a little bit of why I've got all of these accessories here. I have to say that I'm very happy and thankful because a company sent them to me as a present. Yeah, and no conditions, no strings attached to anything. They did not request me to make a video on this. They just sent it to me, and as a thank you, I decided, wow, that's pretty cool. I'm just going to present the things also to you, hoping that they prove to be useful. They are useful, okay? Yeah, as a thank you to the company, I put the links also below into the description, because they are quite practical things here for microscopy, and I'm especially happy about the slides that they sent me and the cover glasses, and there's some sample containers and so on. Okay, so I'm quite happy about that, and I would like to quickly show you a little video first of what was sent to me. Okay, so again, a big thank you to Globe Scientific. That is the name of the company. This is basically a box that arrived, and I didn't know what to expect. And this is basically what was in the box. Yeah, it did feel a little bit like Christmas. There are those centrifugation tubes, which are really practical for water samples. Yeah, when you collect water samples, there are slides. These are slide containers. I'll have a closer look at them later on. Yeah, I have to say something about those slides because they're really nice as well. Yeah, some plastic pipettes were in here. Okay, and yeah, this here also some cover glasses. I did not open them yet, so I'm going to have a look at them together with you. Yeah, and on the bottom there were even some catalogs, and yeah, I did not know that these quite heavy actually. If you also want to download the catalogs, you can download them as PDFs from the website. Again, I put a link into the description below. Again, I would like to thank the company very much, and yes, as a thank you, so to say, I decided I'm simply going to present it, also because I did not look at some of these things myself yet. Okay, so that's basically something that I would like to do. For those of you who are basically new to the format, if you have any questions, please do feel free to ask them. Okay, and off-topic questions I will try to answer later. Every now and then I'm going to look over to the chat to see if there are any questions here. Okay, please. Indicate questions to me with @Oliver or @Microbehunter. Please do a video on stains. Okay, so yes, as a matter of fact, stains. I've got stains here. Stains video is in planning. Okay. Yeah, I just wanted to say this as well, because I did order some stains from another company called now I forgot the name. It's a company in the UK, and Brunel Microscopes. Here it is from Brunel Microscopes in the UK, and they arrived, but they're not at the post office. And due to the weekend, yeah, I could not pick it up, and therefore I'm going to show you probably some staining, hopefully next week. And I also bought some glycerol gelatin to do some mounting of pollen grains, which I also would like to do. Okay, so this is something. This is something that I would like to try to do. So what I'm going to do just for a couple of minutes, I decided is to start unpacking this. I have to admit, one of these are already unpacked. Okay, and that is basically this slide box here. And I just want to show this to you. I already opened it and had a look. And yeah, I don't know if you've already seen those or not, but those slides here are a little bit different than other slides. Look at this, what they have here. Yeah, they've got this. It's not a frosted, but it's yeah, colored here, so you can actually directly label it here using a pen or even a pencil here. And the slides are look when you actually see the reflections. Do you see the reflections here? What do you notice? Look at the reflections of the light. Yeah, some of you might have noticed already. I couldn't believe it. They are clean. They're clean, really. Something that is not always a given. I just want to show you another slide from a cheap slide box. Not slide box, but slide container from Cheap Slides. And these here, let's have a look. Yeah, if you look a little bit here, you don't see that very much. But if you hold it into the, again, you see that there is this water stains on here. Now maybe now you're able to see it. Do you actually see that there's this white? Yeah, and compare this now to the quality one. Yeah, I think maybe if you just look at the reflection, yeah, there is a huge difference here. Yeah, so this basically means that with those clean slides, at least I save the cleaning process. Yeah, so just a short info why these why I was happy that they sent them to me, because they have also those 45-degree angles here, so that you cannot hurt yourself, and also the edges here are how do you call this, polished. So essentially there are there's less chipping, because people who are using those slides in the medical field otherwise might injure themselves, or the rubber gloves might get damaged. Yeah. So for this reason they are quite nice. So I would like to say a big thank you yeah, for having sent them to me. So that's one of the things. One of the things I did not look at yet are those cover glasses. I am running low of cover glasses anyway, so I'm just going to open them. Again, not a sponsored video, okay, but still, yeah, something where I would like to simply present it to you in any case. Let's see how this looks like here. This is a pretty, yeah, that's a pretty solid. It's a pretty solid box. How can I open this? Wow. Here we go. That's look. That's nice. Okay, good. They're useful. And okay, look at this. Look, these are gigantic cover glasses. They're nice. Okay, also clean. Yeah, normally the cover glasses. Look, I just show you normally cover glasses are small like this. Yeah, but these are big ones. That's actually quite good. Okay. I use occasionally the big ones here for if you need to have a very large film, a very large thin film. For example, when I used to put my own blood under the microscope, then you put a cover glass like this on your blood, and then the whole blood spreads over a large area, so you get very thin layers. Yeah. Okay, well, very nice. I'm quite happy with the result. Not the result, but the little present here. I'm going to quickly stop again here and read some of your questions here. You should ask for other promotions. Yeah, okay. And real clean slide, that's probably cost a fortune. Yeah, what I usually do is the following, is I do wash my own slides again. So just to show you, I've got a little plastic container here, and just show it to you. And over here, in this container, I collect all of my slides, and then I wash them, right? And usually with hot water and with dishwashing, yeah, I reuse them because otherwise it would be a little bit of a pity. And generally, I do not observe infective material anyway. I mean, it's just kind of regular stuff, non-dangerous things here. And for this reason, I just clean them and I do not, simply throw them away. So this is something that I just also wanted to show you. Let's put this here, and I go through the questions again. Best type of cloth to use to clean dry slides? I have an idea, yes. Okay, so I'm going to show you the cloth that I use to clean the slides. It's a little bit dirty already, but this one is really nice. It's dirty already, okay? But this is a so-called a microfiber cloth that is used to clean glasses. And why I use this is because those microfiber cloths, they do not leave those fibers behind. You can use, of course, also regular tissue paper, tissue paper, but sometimes there are those fibers left behind. But I found those microfiber cloths to be very, very good. So what I'm going to do is I'm going to replace it because it's already pretty dirty. Right, but I got this from a shop for my eyeglasses, and these are very good because you can dry wipe the slides using a cloth like this. Yeah, let me just have a look here again. Here it is. Yeah, so usually what I do is simply I don't know if you're able to see this. It's still kind of dirty, and then usually I dry wipe it. Sometimes this actually works well, sometimes not so. The water stains can also be easily removed with a little bit of vinegar. Yeah, but those microfiber cloths are very recommended. Yeah, so let me have a look. Let me have a look here. Jesse researched something. Oh wow, 72 of these slides is $53 on the website. Oh, okay. Now these are quite good slides. So big thank you to the company. But what I want to try here is, and that's a little bit something I want to try out is I've got a laser engraver, and with a laser engraver, I'm able to engrave some text over here as well. I don't know yet. Yeah. So let me have a look here. Yeah. Your used slides would dry up after use and become harder to clean. Generally, what you have to do is if you wash your slides, or when I wash my slides, then I have to immediately try to dry them so that essentially there are no water stains. Yeah. So I generally did not find it a bit difficult to clean the slides, and I also tried it over the dishwasher. I put them into the dishwasher, so this also worked. But generally, if you do not allow it to dry completely, then essentially I did not have any problems so far. Okay, so let's have a look at a few other things here that they sent. And I'm going to talk about those containers later on as well, because I like those containers here. Not only for measuring out things here, obviously, but they're very good for storing water samples. I sometimes have stored, and I'm going to show them to you later as well. I've got those, not exactly the same ones, but I like those tubes because I put a water sample in here. I generally do not put a lid on top because I do not want this to turn anaerobic. And then essentially I simply store the water in here, and every time when water evaporates, I'll add a little bit more. These are centrifugation tubes, not very practical in this sense, but I'm going to show you a solution because of course they cannot stand, but I'm going to show you a little thing that I 3D printed. Link to the files also in the description below. I made myself, I'm going to a little stand here, a rack, so that you can essentially also put those things into the rack. Yeah? Again, I simply designed it very quickly a few days ago. If you want to have a look at the files for this rack, then. Yeah? So there are also smaller tubes over here, which essentially can also be used. So usually what I do is when I go out, I usually take always a couple of these tubes along, simply in case I find some samples, water samples or otherwise that essentially that I would like to take along, and then I basically put them in there. So this is basically nothing unusual so far, but I did get some other containers here. Well, okay, these are disposable pipettes. What I usually do with those is I've got also, you've probably already seen it, that I like to use them generally. And what I usually do is I don't throw them away after single use, but I rinse them out. Yeah. And however, those here are a little bit finer than the ones that I have. So you see that they're quite nice, fine. And yeah, also very practical for taking water samples and some also maybe some for catching water fleas and so on. Huh? So, however, those things here are also interesting, and then I would like to move on a little bit. Look, these are also containers. Let me see. Okay, these are also containers. Okay, now a little bit stuck here. Yeah, and in these containers you can put slides. So let me see. So let me. Okay, you can basically slide. Okay, slide transport. Slide transport containers. So in case maybe you want to ship some slides or so, or yeah, carry them somewhere, then they're useful. Okay, so this there are different versions of this, and here as well. Okay. How do I open this? Here it goes. Here it goes. Okay. Yeah. Yeah. You get the idea. Huh? So yes, I just I'm going to put everything back here, and basically they sent me a couple of these slides with different colors, yeah, of the labels here, and yeah, they're quite nice. But I'm probably not going to simply use them for regular observation, but maybe I'm going to use them for slightly better. Maybe I'm going to use them for making permanent slides or something like that. Just for your information, the reason why those slides have polished edges and 45-degree corners is because in medical laboratories where you're working with infectious material, you have to be careful that you don't hurt yourself. Otherwise, there's the danger of infection. Okay, and that's why these are basically safety aspects here. Okay, so yes, these containers are great for modern slides. Many old antique slides won't fit in them, sadly. Ah, yes, that's a good point. I've with some of the antique slides that I also bought, they are too thick. Okay, and I even had some problems fitting antique slides into regular slide boxes. Yeah, because I think some of the old slides are I don't know, I would say about this thick. Yeah. So, and then of course it won't fit in there. Yeah? What percentage of the container do I fill up with samples to keep them alive? That's an important question. Okay, so I'm going to show you something here. And so the question is how much should you fill it up? Okay, careful. I'm going to show you now something, what I've got here, and then I'm going to try to answer your question. Okay, so here look what I've got here. So this is, yeah, these are also some samples that I would like to look at. And this is a 3D printed rack that I made. It was printing for quite a long time. Again, I put the files into the description below. There are 3 files. And yeah, I store my water containers here. And look, what I've got here is this one here I closed off. And it's been in here now for, I don't know, weeks or months. And if there is lots of air like this, the majority air, it's not a problem. A couple of years ago, I made a mistake. Okay, I went on a holiday. I took along a lot of these containers to collect water samples. And basically, I filled up the container all the way to the top. Yeah, and then I closed it. And because I wanted to take along as much sample as possible, and after 2 or 3 days, it already started to smell sulfur, like sulfury, hydrogen sulfide, foul. It smelled foul. And that's a bad sign. It means it went anaerobic, the microorganisms die. But if you keep a lot of air, then you can keep it for, I don't know, for days, weeks even. Huh? Of course, every now and then I open it up, I exchange the air a little bit, yeah, by going like this, yeah, and then it's going to be fine. Huh? But I found that if you have very little water in here, I had some worms there as well, and I was able to keep them for quite a long time. Yeah, but I would say definitely more than half should be air, yeah, and then it's going to be fine. You see that the containers over here, I keep open. This, of course, means that water is going to evaporate, and then I always add a little bit of water to keep the sample going. And I found some interesting, more or less interesting microorganisms in here. And those samples here are several months old already, so that's a little bit my my collection. And I'm going to look at those here also today. Look, this one here as well. This one, yeah, this is not a water. These are some leaves in alcohol. Yeah. So you see that, yeah, that's why it has to be closed. This one over here is a water plant. Yeah, but again, you see that, yeah, it's marine water, salt water. So I closed it so it does not evaporate. It's yeah, it's not the ideal way of, it's not an ideal way of actually storing samples like this, but it's a little bit experimental here. Yeah. Here as well, you see a lot of biofilm forming, yeah? And every now and then I add some more water on top. Yeah? So that's basically, yeah, the thing. Just make sure that the sample does not go anaerobic. So let me have a look here. Do you have an email to contact you? Yes, it's. I've got several emails. editor@microbehunter.com. Or you can also send it to oliver@microbehunter.com. Yeah. I'm going to go through here. Too late or too early for the autumn skeleton leaf video? Yes. I'm going to just check if there are any comments from me. Okay. That's why it does take. Yeah. Okay. So there's a small correction. 144 slides for $57. Yeah, but again, these are I would say special purpose slides a little bit with the color here. Yeah, so in any case, what do I want to do now? I just want to show you very briefly the 3D printed project here. If you're kind of wondering why I've got so many colors here, blue and white, this is because I used the remaining filament of my 3D printer. Yeah, and yeah, basically I just made some holes here, and I glued it together with nail polish, clear nail polish that you put in your fingernails, because this nail polish contains a solvent that also softens the plastic a little bit. Yeah, and this design actually works quite well, and then you can basically put your containers in here. Okay, yeah. So just saying that this is basically something that, yeah, and those holes here on the bottom is are basically so that you are able, yeah, those with yeah, that have basically no flat bottom, that they're also a little bit stable in the center. Yeah? So just wanted to share this also with you a little bit in case it proves useful, or you can make stuff like this maybe out of cardboard. So just saying, yeah, let's have a basically let's do a little bit of again a little bit of biology. I also want to show you now how my slime mold cultures are developing. Let me put this. No, I'm just going to put it over here a little bit because I will need them again. Yeah, by the way, I want to show you something else here, which I got from, which I borrowed from the lab. I don't know if you know what this is here. Okay, this is a so-called made of glass. It's a staining container. Let me take this out here, and you can basically put the slides in here for staining. But that's not what I want to do. What I would like to do is I want to fill this up with pond water. Okay, and I would like to leave the slides in here because I would like to grow a biofilm on those slides. And then I would like to put, yeah, basically a biofilm means there are algae then growing on this, directly on the slide surface. And then I would like to put that under the microscope, okay? Because when you scratch off a biofilm from somewhere, then you're damaging the structure. But by simply growing it directly on the slide, I would like to simply observe how it looks like. So that's basically the reason. But these containers are actually used for staining, so that you can basically, if you want to stain a slide, you put it in here and then you take it out again. So just saying. And the reason why I clamped this together here is the following. There are two slides. Because this way I hope to only be able to grow the biofilm on the outside surface, so that basically the inside surface is free of a biofilm, so that I can put it more easily on the microscope. This is kind of was a little bit of my idea. I didn't start yet with that, but that's going to be a long-term project. because it's probably going to take several weeks or months or so for the biofilm to grow. So what I might be doing also is I'm thinking also of adding maybe a little bit of a drop of plant fertilizer maybe to promote the growth of the algae. Yeah? So that's something I've not tried before, but I just wanted to share it with you as one of the projects that I've planned. Yeah. So let me quickly have a look here. So that was an email question. Okay. Yeah. I will have here. Yeah, you will get a biofilm on both surfaces. Be careful to not mess up your scope. That's correct. Yes, that's correct. That's the reason why I was kind of thinking about clamping it together so that I have only a biofilm forming on one surface. This was kind of a little bit the how do you say, the idea. But I have to see how this works. So, of course, I'm aware that the bottom surface of the slide must be clean, otherwise I'm going to really mess up the scope. I definitely don't want that. Yeah, thank you for reminding me, but this is actually indeed something that I've been also worried about. Yeah. So this was from a comment from Alexandre. Yeah. Thank you for the cardboard version idea. A lot of us are not advanced amateurs and don't have 3D printers. I might also try cutting into plastic. Yes, that's definitely a possibility because those things do fall over. I mean, even if they have a flat bottom, if you just keep it standing over here, you won't believe it how often I had a mess already because they were tipping over, right? So cardboard is definitely a possible way to go here. Or maybe, I don't know, if a wire system, I don't know. Yeah. What I've also done is the following, and this is not so nice. Yeah, you take it and then you can do it like this. I've done this a lot. Yeah, but then I said no, it was annoying, and then I decided to 3D print a little bit. It took me a full day, I have to admit, because yeah, I had some issues with a 3D printer. Yeah. Why not put the slides on a string directly into the pond? Yes, I could do that, but it's not practical enough for me. That's it. Yeah. Does the dry chlorella algae tablets come back to life and grow when you put them into water? That's I didn't try that yet. Yeah, I've got these. Just a second. This is basically these tablets that you see here. These are chlorella algae. And you know what? I could put them also under the microscope. They are sold as I don't know, people eat them for, I don't know, health reasons. I use those algae to feed my water fleas. Yeah, that's dry yeast here. But I don't know if they actually come back to life. Yeah, I would have a hard time imagining, but who knows? Yeah. So I'm going to quickly read some of the questions here. That's, by the way, this is how that's the slime mold, in case you were wondering. Okay, I will tell you why it looks very dried up, and that's deliberate. Okay, we could put tape on one surface, yes, for the slide. The 3D suggestion, the tape, yeah. Okay, box of sand, yes. Will you use cover glasses for those biofilm slides? So generally, if you have a biofilm growing, let me put this again over here. Yeah. And let's say that it's really only growing on the top surface. Okay, the bottom surface should be clean, or maybe you just scratch it off, or yeah. Then if the magnification is low, then essentially you do not need a cover glass, like 4 times or something. But generally I still would put a cover glass on it simply to make sure that the water liquid is flat, and also because I want to protect my objectives. Otherwise there might be the danger that I'm going to get my objectives wet. But then for those slides, I could then imagine maybe putting on those large cover glasses. Yeah. So another question here. You can put a thin layer of silicone on one side of the slide and it will be waterproof and easy to take. Ah, that's another one. Interesting. Thank you. So basically you put a thin layer of silicone and then you peel it off. Yeah. Again, I have to try. Yeah. So I would like to simply show you this here. I do have a couple of other slides, not slides, Petri dishes as well. You see I've been trying a little bit of staining here. And the behavior of those slime molds is quite interesting. Okay, I'll first talk a little bit about this here. So what I've done is I've grown. Let me show this to you. And then what I've done is these are some oats cereals like those here, right? Breakfast oats, and I've grown the slime mold, on those oats, and this was basically completely yellow. And then after a couple of days, look what happened. It started to spread. So basically, it moved away here, and it even tried to escape here. Look on the side here of the petri dish, right? And it's now slowly started. The filter paper is now slowly starting to dry. This is deliberate because what I would like to do is I would like to then when it's dry, you're able to store it. Yeah. So the slime mold is able to survive dryness, and then when you cut those basically the filter paper into small pieces, then you can store them, and then when you put them back into moisture, into water, then it comes back to life. And this is kind of what I would like to try. The reason why it moves away, and you can also see it very nicely over here. Look, this is for filter paper. This one is using agar. Yeah? You can actually see, this is basically, I was trying with some staining here, so ignore this blue spot. But you see how the slime mold actually started to move away from the cereal like this. And what I think is that what happens is that the slime mold will actually accumulate wastes and release wastes, and it's usually the pH drops. It's usually an acid and other things that these microorganisms produce, and I suppose there is a pH gradient. That's why it knows how to move into which direction it has to move to look for new food. So it kind of tries to escape its own waste, and therefore it kind of goes down the concentration gradient. Yeah, so that's why you see that it's kind of starting over the days, starting to move outwards here. Yeah, and this is also what happened over here. Yeah, the slime mold started to move away from the cereals. Yeah, and outwards. And and over here as well, yeah. You can see that, yeah. And it seems to be like this, that it's kind of supplied with new food. There's always something kind of connecting it back to the food, yeah. And then new food is kind of, or nutrients are being pulled forward, and as the front part tries to find new food here. So what I would like to do now is the following. I'm going to put this under my stereo microscope, and then I'm going to show you something else that I've done. I don't know, maybe many of you don't have a stereo microscope, but doesn't matter. I still want to show you something. Okay, let's try to focus this. Okay, there's the arrow over here, which is a little bit annoying in this case. So here we go, and let me see if I'm able to do that. Let me see. I'm going to rotate. Why does it become dark? You might wonder. And then it becomes bright again. Okay. And look at the following. It looks a little bit, you're able to see maybe, just look. This looks kind of a little bit out of focus, and this one over here looks a little bit in focus. Okay. Which is normal because there's a certain depth of field. So this one is clear of the slime mold, and here because the oats here is higher up, therefore it's a little bit blurry. But look, I'm going to turn it back now. I'm going to turn it back. No, other way. And it's not very strong, the effect here. Let me go in a little bit further. Have to, maybe that's the one. Let me rotate again. I don't know, the effect is a little bit stronger. Yes, that's the one here. The effect is a little bit, I don't know if you're able to see it. Now it's like this, that this one is a little bit, this is a little bit in focus, and this one is a little bit in focus. And when I turn this over, then you see that it's more blurry. Okay, I need to explain what I'm doing here, okay? The thing is the following. This was in my other project that I've done. And I tried to reduce the aperture of the stereo microscope to increase the depth of field. I'll show you what I've made. Do you see these things here? Yeah. Also 3D printed. And I rotated this, and basically I decreased the diameter of the lens, so to say, or of the aperture. And this basically gives me a greater depth of field of my stereo microscope. Yeah? So of course there's less light coming in and so on, but actually, and I was rotating them, yeah, around like this, yeah. So large aperture means more light, but lower depth of field, and then by rotating this one in place with a smaller, so the lens is much bigger than this hole, and therefore yeah, the depth of field is larger, and then basically you can look at thicker specimens and basically more parts are in focus at the same time when you decrease the aperture. Yeah? So I just wanted to, I'm still playing around with this a little bit, okay? But in case some of you have a stereo microscope and you would also like to cut some holes into some cardboard and put it, under the stereo microscope, then you can also play around a little bit by increasing the depth of field. So it's basically also what you do in photography to adjust the depth of field. Just also wanted to share this with you in case you're interested. Yeah. Yeah, some questions here. What's better to use, the filter paper or agar? Honestly, it depends. I've got success with both of them, and I would say filter paper is way easier to use, of course, because you don't have to make the agar. But I also but it dries up faster, obviously, because the agar contains way more water. Yeah, you can see how thick it is, and therefore it stays moist for much longer time. Yeah? But if you want to actually dry it, then of course you have to use filter paper. Yeah? So, and what I'm going to basically, let's just have another look at this under the stereo microscope. So you see that they have grown over the last couple of days quite a bit. Yeah? I think we just reduce the. Yeah. You can see also here at the top that there is, I'm already reaching the edge of the, I also have to rotate this back to the center. Yeah. So here we go. Huh? Those slime molds are really fascinating creatures. Yeah? So what I have not, and then I would like to look at some of the water samples here. What I was not so successful with, you see maybe this here I put some oats into crystal violet. I stained the oats, kind of hoping that the slime mold is going to take up the pigment, the stain. It didn't do that. It actually tried to avoid it. It actually moved away a little bit. Yeah. So yeah. So you see that this is not quite as successful as I hoped it to be, because I was kind of hoping that this way I'm able to change the color of the slime mold. Yeah. So you see all work in progress. Yeah. Lots of bacteria here. So not very nice. You got to be a little bit careful. Yeah. So, but the slime mold likes to eat those bacteria in any case. Yeah. Yeah, I'm reading again. Yeah, could be a nice video theme for Halloween, huh? Why is the screen with the slime mold a little bluish? I don't know which one it was. It could be maybe it could be that the how do you say the camera did some incorrect white balance. Yeah. Well, this blue thing here, in any case, this is the crystal violet stain. Yeah. And maybe it does look a little bit bluish because of the of this background here. By the way, another thing, I'm just sharing a couple of projects. This here is black plastic, but you see it's actually not really totally black, but it's kind of grayish. There's this color called Vanta Black, which is apparently the blackest black that's paint that's possible. So I think that's also one of the things that I would like to try. Yeah? That to get the background completely black and dark. Yeah. So where is this? No, that's the wrong. The desk is here. Okay. Yeah. So there's some off-topic questions that I would like to ask later. Okay. The depth of field or aperture. Yes, I've been throwing around some. How do you say, some photography terminology that not everyone might be familiar with. What I basically in photography, aperture is referred to as the opening hole through which light is able to go. And this is for the stereo microscope. There are two objective lenses, one left, one right. right? And I've kind of reduced the hole through which light is able to pass through, right? And when you do that, then of course less light is able to reach, yeah, your eyes. It's going to become darker. But as a side effect, what's going to happen is that thicker specimens, the top and the bottom, will be basically in focus. I give you an example here. Let's just use this camera as an example. When I hold my finger up here, then you're going to see that the bottom is, of course, the desk is in focus, but my fingers are blurry. But if I somehow were able to change the opening of the lens of this camera, if I were to make it smaller, then the finger would also go into focus, both of them. Yeah, and that's basically the depth of field. Yeah, basically how far an image is going to be in focus. Yeah, and this was one of the things that I was kind of trying to play around with my stereo microscope. That I wanted to simply increase this area so that the higher parts and the lower parts are both in focus. Yeah, and you can do that by decreasing the opening here, and that's a little bit the experiment that I've been playing around with. Yeah, so just some ideas here. Yeah, yeah. So very quickly, you know what? Yeah, I'm going to now do the following. I'm just I want to do now a little bit of actual microscopy as well. So I would like to put some of those water samples here also under the microscope. So I'm going to put the Petri dishes somehow away. Always also checking a little bit also the time. So let's turn this off here. Let's put this here, and let's, you know what I'm gonna do? Just because I've not tried this yet recently, why not try to put some of this chlorella algae under the microscope? Yeah. So I'm just a second. I'm gonna show you how it actually came. Some time ago, I bought. Yeah, that's how it was sold. Yeah, I bought this over Amazon because I simply needed some food for my water fleas and so on. Yeah, yeah. So this is how it looks like. Yeah? Seems to be some kind of a food supplement. So apparently, I don't know if they're healthy or not. But if you just look at it, you see quite a few of these tablets. Yeah, compressed algae. And these are the ones that I actually am using to feed my water fleas. And what we can try now is we can actually, I need now a little bit of water in here. Up. Now I really made a mess. Okay. And I'm going to scratch off some of this here because there was the question, are they still alive or not? And I have to tell you, I don't know. And I mean, one would probably have to wait a little bit and see whether they start to photosynthesize. And when I feed my water fleas, I usually take a small amount like this, yeah. But let's put it under the microscope simply to check. My pipette is here. Yeah, you draw it in and out, yeah. And let's see how this if this actually works or not. And it's a little, I think maybe a little too much water. Yeah, because if there's too much water, then you cannot go up with the magnification quite as much. Oh, yeah, this is a very, somewhat improvised way. So let's have a look at these, because, yeah, I want to do a little bit of, so I need to change this. Where is this? Here we, here we go. And let's put it up in here. And this is how it looks like. It's, you know what, I'm gonna put the filter away. Still see a whole bunch of clusters. So what I probably am going to do is I'm just going to maybe dissolve a tablet in a little bit of water and wait maybe a day or so, and then see, put a small sample under the microscope and see if photosynthesis happens by watching air bubbles. Yeah, air bubbles grow. Yeah, but that's basically the tablet. So I don't know if they're still alive or not. Probably not. I would say because they were dried, but who knows? Yeah. So I find myself a little bad after looking at the wonderful microorganisms to just let them die, throwing them in the 70% alcohol solution. It's a little bit absurd, I know. Yes, honestly, it's a very common thing. Is you look at the microorganisms, maybe tardigrades even, maybe even some cute ones, and then essentially what do? You kind of either flush it in tap water, or you disinfect it or so, or you simply let them dry up. That's essentially what I'm doing. I put them into the plastic container, they dry up and they die. But I think what's really important is to understand is that many of these organisms, they do not have an individuality just as we know it, we humans have. You don't feel sad about your skin cells either when you scratch yourself, right? Yeah, you're killing off thousands of cells this way, right? But there is no personal connection there, right? And for this reason, I think what we have to do because they're very common, the ethics aspects are very common. What we have to be careful is if we want to be ethical about, protecting nature and the environment, there are some real issues that have to be dealt with. And I think with microorganisms, that's fine. Yeah, let's not feel too sorry about them. We are respecting them in any case because we observe them under the microscope, and we like nature for that reason. But I think we have to stay in that sense realistic, because every time when I eat yogurt, I love eating yogurt or cheese. Oh, beautiful. Yeah, cheese contains fungi. Yogurt contains bacteria, right? So essentially we kill billions of these cells every time when we eat something like that. Yeah. So, but I get it. I mean, and I have to be honest with you, every time when I look at some of the microorganisms, especially the larger ones, like, I don't know, water fleas, for example, or tardigrades or whatever, I usually always put them back into the jar. Yeah. So what we're going to do now is I'm going to show you here where is the desk view. Yeah. So it's I have to tell you the following. I made a little mistake here. I was very sloppy. Every time when you collect a water sample, you should do two things. You put the date on it and you put the place on it where you collected it. I did neither. I have no idea. I don't remember anymore. I don't remember when I collected them, and I don't know from where I have them. I know. That's not the best way. And some of them are quite old already. What have been quite old? Several months. Yeah. And I always kept on adding a little bit more tap water every time when the water evaporated. And of course, what you have is you have a progression of life in there. This means that over time it changes. Yeah, depends on the not only on temperature but on the amount of light that there is, and so on. Yeah. So the question is now, how are we going to get this out? Huh? I mean, there is some solid material in here as well. And I will tell you that I usually use either a pipette or tweezers every now and then. Yeah, every now and then I'm going to read the comments. Yeah, you keep Microbehunter, you keep forgetting to label things. Naughty. Yes, that's sloppiness. Yeah, that's why I always label your things. Yeah, look, this is just before I actually So that's all that we need. And I'm running out of cover glasses as well now. This is very little, and I usually press it apart a little bit, and let's have a look at this. This slide here, for example, with the chlorella algae goes now into my plastic container. And let's have a look now here. I always also forget to switch over. Okay, so Yeah, and what do we see here? Nematode worms. Yeah, again, this is a pretty old water sample, maybe over half a year old maybe. And I'm always fascinated to see how much life there is still present. Lots of diatoms, nematode worms, and so on and so on. So what does this tell us? If you basically keep the container open and yeah, if there's an exchange of air possible, if you always keep on filling in a little bit of water when water evaporates, yeah, chances are pretty good that the water sample will stay alive. Yeah, of course. Where was this again? I just had it and I forgot about. Yeah, this looks. Let me see. Yeah, this is something. This is something that you see these days quite often. Okay, it could be. I don't want to be too fast, but these are fibers here, right? It could be natural fibers that basically occur, I don't know, cellulose fibers, but in many cases, these are also microplastics. Yeah, so almost unavoidable these days. Yeah? So many synthetic fibers from clothing are microplastics. You find them a lot in water samples. Those things here in the back, those bright ones, these are cyanobacteria. Yeah, probably oscillatoria. Oh my gosh, thank you very much. I just saw that there's a donation here. Okay, thank you very much. Yeah, and I'm going. Yeah, I'm going to show you just a second. I've got another slide box here because with the donations that I receive, I buy slides, and I bought recently a slide box. Yeah, and yeah, so these are lots of cyanobacteria here as well. Let's go up with the magnification. Yeah, and yeah, so let's talk a little bit about, and those cyanobacteria, those oscillatoria, they have a gliding movement. If you actually look at it for some time, then you might see that some of them actually move slowly. Now this one not so much, but, yeah. So a short little story that I was told by one of my university professors long time ago about, these are of course the nematode worms, about those cyanobacteria, because those cyanobacteria, those blue-green algae, are actually bacteria. They do photosynthesis, but they are bacteria. They're not really algae, but they were traditionally called blue-green algae. Yeah, and the story goes as such. Actually, a sad story. Yeah, but it's one of our professors told it to us that one of his professors, this must have been already a long time ago. Yeah, essentially, they discovered that those cyanobacteria are not blue-green algae, but they are actually bacteria, something totally different. Yeah. But at that time, biologists classified organisms to a large extent also based on their appearance. They're green, they're living in water, they do photosynthesis, so basically they must be algae, right? It's almost like saying that all green cars are, I don't know, BMWs. Yeah, nobody would say that, right? But a little bit, this was what taxonomists and biologists did many years ago. They kind of looked at it. They said, look, lives in water, does photosynthesis. Yeah, looks like a filamentous algae, must be algae, right? But actually, it has nothing to do with that. And this professor, my professor's professor, when it turned out that essentially that these are actually not algae but bacteria, he was massively upset about that. So upset that he actually got ill. And when this by the way looks a little bit like I don't know what this is, yeah. And he was totally upset about that. When we heard that story as students sitting in the lecture hall, we had a kind of laughing at that when this story was told to us. This is not a laughing matter, because it shows that science progresses, and that sometimes people who've been studying something for their whole life, and then when they discover that actually they were completely on the wrong track because of new scientific discoveries, some people take this quite personally. Yeah, and it looks a little bit like a rotifer here, this thing that's moving. Yeah, but in any case. those blue-green algae are bacteria because they do not have a nucleus. Yeah? And they are indeed responsible for the production of a significant percentage of the world's oxygen. Yeah? Yeah, and a whole bunch of worms again. Yeah. So I'm going to quickly have a look again at some of your comments. Will UV illumination show the microplastics better? Yes, if you stain the microplastics with a pigment called, or a dye called Nile red. A Nile red is a hydrophobic dye which will stick to the plastic, and then when you use UV light, it should actually shine up. Yeah. So I don't think that the microplastic itself will start to shine up with UV light, but you have to stain it first. And when you do that, then you should be able to identify it without problems. Yeah. Cyanobacteria are large compared to normal bacteria. Good point. Good point. But I'm just going to show you. Maybe not. So let me see if I find a good specimen here, because what you see here is not one bacterium, but actually many of them. Yes, see, this one here is gliding. Yeah. See, these are not worms, but these are cyanobacteria again. So let me open this. Yeah. So what do you have here? Yeah. Here you see the individual cyanobacterial cells. Okay. So that's not one bacterium here. Yeah. But actually these are many cells in a chain together. Yeah, you see it also over here. Yeah, so in that sense, it's nematode worm, quite nice. Yeah, so and in some species, what you have is you see that there are some cells. I've seen this also once. Some of the cells are actually different from the remaining cells. These cells are called heterocysts, and are specialized for so-called nitrogen fixation. And this is interesting for biologists because it actually shows that there is a very, very simple form of cell differentiation already happening. Cell differentiation means that there is a division of labor. Look at this nematode worm here, right? Yeah. But the individual cells of the cyanobacteria are those little things that you see here. Yeah, yeah. So this is not one cell here, but actually many of them. Yeah. By the way, in time lapse, this looks pretty nice because it looks like those cyanobacteria are actually moving around like worms. Yeah, but here, that's a real worm. What you have here? Yeah, it's a nematode. Yeah. So over here as well. Yeah. Here, this one here, it's actually kind of yeah, interesting. Yeah. So you see that there are also the individual cells in here in this cyanobacterium. Those things that are moving, these are diatoms. Yeah. Yeah. So, and these over here are eukaryotic green algae. Look, there's another. What is this here? It's another one here. This is also a diatom. Yeah. Yeah. So they have a silica shell. Yeah. Yeah. So very quickly. I hope I'm not forgetting any of the questions here. Okay, yeah. Yes. You know what? What time is it? I'm going to have a look at another one. Actually, I said we're just going to look at all 10 of them, but who cares? This one over here, that's not one. I don't know about this here. Yeah. How am I going to do that? I'll switch. I'll use different tweezers. I mean, I could use the same tweezers really, but on the other hand, I don't want to cross-contaminate. Yeah, so I'm going to just take. I mean, they're not sterile anyway, but those here, what you see here, are normally eukaryotic algae. Those here maybe not so much anymore. Let's have a look. So it's usually because I'm in Central Europe. Here it's of course like in other countries winter time. I just visited a local pond the other day, but the water was very clear. Not many floating, not much stuff floating around. So it is not a lot of water here. So I'm going to add a little more here. So here we go. Just a second. Let me redistribute. Yes. Ah, yeah, something is coming. Thank you very much. So I got again your questions. Okay, that I can respond to. Maybe many of them I might have already responded to, but yeah, I've got. My son is helping me because he's copy pasting your questions onto a piece of paper, so it's easier for me actually to respond to them. So let's now basically, where is the scope view? Here we go. Let's turn off the arrow again. It's way too bright, way too bright, way too bright. That's basically another sample. You see, one of the reasons why I want to show it to you, because it actually shows how different they are. Yeah. You see, there's lots of tiny stuff, maybe some bacteria, maybe not. They're pretty large for bacteria actually. Yeah. Yeah. You see over here again, less nematode worms. Yeah, more smaller microorganisms. Let's go up. Some magnification. It's now bright field. You know what? Let's put this here, in here. Yeah. Maybe not quite as exciting as the other one. Yeah. But I mean, you get the idea. Each sample has its own characteristics. Yeah. So let's move up yet further. Yeah. I mean, you get the idea. Huh? What is this here? Yeah. Still interesting, but they're fairly small. Yeah. Ah, yeah. There's a little air bubble kind of burst, and that's why there's this water stream. Yeah. Yeah. So, yeah. Let me see. Yeah. Those algae here also don't look particularly nice anymore. They look kind of a little bit decomposed already. Okay, let's have a look. I don't know, maybe there's another one over here. What time is it? Yeah, this one actually is. Yeah, let's have a look at this here. I have not looked at this one yet. Let's remove this. And where is the desk view? Okay, and yeah, now I'm going to take some of the scissors here to take a sample, because I always want to take some fresh tools. This is way too much. Okay, I'll take a pipette and let's do the following, because this is definitely too much. So sometimes it helps to kind of wash out a little bit whatever you've got in here. Okay, and I don't know if this works or not. Let me see. There's not a lot here. So I also have to make sure that you only get one cover glass. And way too much water again. The reason why I occasionally check, because I do want to replace those water samples again, especially when springtime comes. I want to get some fresh samples. And so every now and then I have to throw out the old ones. So, oh yeah, this one does not look to be particularly interesting. Where is, maybe I've got the focus wrong. Here we go. Yep. Yeah, this I simply got too few. Yeah, this one is not a very interesting one here. Yeah. So I think I'm just going to not. Yeah, Generally, if you don't find something interesting right away, then just change change samples because yeah. So, but maybe this one over here from before. Yeah, this was the one from before again. Yeah, there's way more going on here. Yeah. So what I'm gonna do now is a very quick look at your comments. A joke I found in a YouTube comment somewhere: musicians don't try to just decompose. Ahaha. Yeah, pun. Uh-huh. You don't have nice slides. You don't have the demonstration problem today, at least not yet. Yeah. Any tip to collect dust mites from bed sheets about swabbing the area? Not swabbing, but you use tape or vacuum cleaner. Maybe filter paper. So basically, I've not tried yet dust mites, but what you can try to do is you can, of course, you can try to vacuum clean it, or what some people have also suggested is that you take some sticky tape and that you basically collect them this way. If you want to isolate insects that are on sticky tape, then I sometimes found it difficult to get them off using water. So sometimes I try to put a little bit of water drop on the tape and to pick it off. This didn't always work well. So what you can try to do, because of the glue, so what you can try to do is you can try to add a little bit of oil, kitchen oil, vegetable oil, and this will actually kind of make the insect or the mites or whatever, arthropods, tiny spiders, not stick anymore because the oil kind of dissolves the glue a little bit. So I found it better for picking that off than using water. So yeah, I'm going to do the following now. As always, let me quickly check what time is Is there more questions here? Where can we get the less usual microorganism, Vorticella or else? Let's put it this way. And there has also been a comment. There has also been a comment about how do you say about Hydra and so on. Generally, not always, but generally, what I found: if you keep water samples in a container like this, okay, they're nice and practical, but the biodiversity is generally or very often I found higher when you take a fresh sample. So what you do is you go out to a pond and you collect some, not water itself so much, but some sediment from the bottom of the pond, some solid material. And I generally found that in these areas, the fresh samples contain quite a bit of a higher biodiversity. Very often what happens here is in those plastic containers, very often you have some kind of a dominant or a few dominant species. You have a progression, and then sometimes those that were dominant at the beginning, they die off and the new ones come. And then sometimes I've seen that there are some of the samples are very, I would say, boring in the sense that there's not a lot of variation in there, right? So especially if you wait a little bit longer. So for this reason, the interesting ones sometimes can actually be collected directly from nature. What you can, however, try to do is the following. But you have to be really careful that you don't overfeed. I mean, there's lots of organic material in here anyway. But if you really want to have a high concentration of certain microorganisms, you might want to feed them a little bit with, let's say, one of those cereals here that I have. So what you do is you put a small piece in here, and then you wait a little bit, a few days, and then you take that out and you put that under the microscope. But the problem with that is by enriching them this way, you also got to be a little bit careful, because then you have very rapid bacterial growth happening, right? And those bacteria will then overgrow everything. They will push the oxygen concentration down, and then the other microorganisms are also not able to survive. So this is all a very delicate balance with a lot of trial and error. What I am doing here is I just let it stand, and because the samples were different at the beginning, yeah, basically they develop differently, as we've seen here. Some of them still contain nematode worms, others don't. Others contain more diatoms. Some others yet contain more cyanobacteria. So there is quite a bit of how do you say? Yeah, they develop into different directions a little bit. Yeah. So let me quickly read again. Let me read. There is nothing for me up to this point. Yeah. So yeah, another thing, comment because it relates to this a bit. Some people ask me also, they've been looking for tardigrades, for example, so-called moss piglets, and they didn't find any. And generally, what I also found is that the moss that's growing inside a forest contains much more of these. So sometimes the moss is exposed on a rock and growing there or on a wall. And I found that on those exposed moss samples, there are far fewer tardigrades than, for example, moss growing in a forest in shade, let's say in a moist area. So the place where you collect the sample can have a significant, or does often have a significant impact as well. Yeah? So yeah, just saying. What I'll do is following, I'll quickly have a look at some of your questions. Maybe I already answered some of those. Yeah. Yeah. How are the slime molds doing? Yeah, as I mentioned. Yeah, they are growing fine and slowly escaping as well, or trying to escape. What's going to happen to those? On the side, they're probably going to dry up. Yeah. And what I'm going to do is when the filter paper is dry, I'm going to also cut up the filter paper, and I'm going to see how long I can store those. Yeah? If you're interested in slime molds, I bought them. You can buy them as well. Yeah. Do a video of staining techniques and where and what to use them on. Yes, I did buy now a few stains as well in powdered form, which arrived, and I hope that maybe next week or in 2 weeks that I'm able to do a little bit with that. You should also ask, yeah, another promotion. Yes, yeah. It's about the microscope slides. Yeah, I use this microfiber cloth. What type of cloth? Microfiber is the one that basically I prefer because it doesn't make a lot of how do you call these fibers? Yeah. So the means if I use tissue paper, then you have those tissue paper cellulose fibers on the slide. Yeah. So what sort of piece of differential between dirty and clean slides? What sort of price difference? Yeah. Let's put it this way. Those slides here, those clean slides that I have here that were sent to me are not necessarily more expensive alone because they're clean, but also because they have this colored side here for writing. So these are indeed very, and they have also polished edges. So that makes the slides more expensive as well. Yeah, I would say the really cheap ones that I have here, which are dirty, what I do is I simply wash them with a little bit of vinegar to remove the water stains, and then they're also fine. However, yeah, they do have they're much sharp. They do have much sharper edges, and there is the danger of chipping. Yeah, that's why, especially in the medical field, they're not quite as commonly used as maybe as those slides that are basically safer. Yeah? So let's have a look. Is methylene blue, according to Kuhne, I don't know this guy, could be used as a plain stain after fixing like loafer, or should it be executed in a more complex procedure? Don't know how to answer this question, I'll be honest. My experience generally with staining is the following. It depends. It's not only the stain, but it's always in combination with the specimen. So, for example, I tried to stain my nematode worms, the so-called the vinegar eels, using crystal violet, and they would not accept the stain at all. Yeah? So it depends really on the specimen itself and on whether it's going to accept the stain or not. Yeah. So I would say that I would not be able to answer this question as it is here. Yeah. How do I collect sand samples to observe life? Do I need to collect specific area? Sand samples, if you are interested, you are probably interested in the so-called the interstitial spaces. So essentially what you do is there is not really a lot, the question is, are you more interested in the sand grains or are you more interested in the life between the sand grains? And what I would suggest is just do it the way that I've done. Is you get yourself a jar of water, yeah, and what you do is you collect the sand in the so-called the intertidal zone. So you do not want to collect sand samples that have been exposed to the hot sun. That's going to kill off quite a lot of life, yeah, because it can be hot sand can be extremely hot, right? So you want to collect it from there where it's basically where it doesn't dry up so much, right? And this is basically where I collected it, and there will be hopefully then between the sand grains living things, and if you put it directly under the microscope, you're probably not going to see it so much. But yes, I almost forgot, because I put this here also under the microscope. Look, I just made this video over here just before I started the live stream, because this sand sample is from 2 years ago, 2022, 2.5 years ago, on my holidays, northern Germany. Basically they have this in German. It's called Wattenmeer. It's the large tidal zones there. I collected this and I put it under the microscope, and you think that nothing happens. But look, I'm going to speed it up now because I can actually speed up the video. I can speed up the video, and I'm not sure if you're able to see. Look, there is still some movement. I'm going forwards and backwards. Do you see actually there in this top center there is this, yeah? There are still microorganisms in here that have survived. Yeah, and basically I'm now moving forward and backward, right? Yeah, so basically over 2 years. I've seen a lot of gliding diatoms in the sample, yeah, before, and as a matter of fact, here is one. I found one here, yeah. So basically, this is stuff that I was able to find in the sand. Right, and it was actually able to survive in this jar for a couple of years now. I put always a lid on here to prevent evaporation because it's salt water, and when salt water evaporates, then the salt concentration goes up. So essentially, what I've done is I've only kept it on here. In here, and see it's already collected some dust. Yeah, here, yeah, again lots of air. That's important, and a little bit of water and sand on the bottom. Yeah, so yeah. See that this was able to keep for quite some time. Huh? So let me see. Where did I leave off? Your used slides would dry up after use and become harder to clean. Yeah, a little bit. Indeed, yeah, they become a little bit, yes, because they dry up and so on. So what I do is I soak them in hot water for half an hour, usually, yeah. And if they are really difficult to clean, then I just throw them away. Yeah? Can you use ultrasonic machines to clean slides and cover glasses? Yes, you could. And I actually once bought an ultrasonic machine, but I sent it back because I couldn't get it to work. The machine had a defect. But generally, it's like this, that because it's a liquid anyway that I put on the slide, it usually can be washed off quite easily. You don't need a lot of force to clean them. Sometimes I also added some dishwashing powder. Yeah, this also works. Yeah. And simply by rinsing it with hot water, it works quite well. Yeah, and water stains can be removed with a little bit of vinegar. Yeah. Okay. These are the tubes, the SWAT. Okay. Yeah. Yeah, these tubes can be used for all sorts of things. Yeah, those containers are great for modern slides. Many old antique slides won't fit. Yes, we're talking about those plastic containers. Yeah, because some antique slides are fairly thick, actually. Yeah. What percentage of the container? Just I would say 70% of the container should be air, and then it should be fine. Yeah. I wonder what these frosted ends on the microscope slide is for. Yes. Oh, did I not explain this? This is so that you can directly label them. Yeah, you can actually directly write something on here to label the slide. Yeah, so this is basically used also in medical diagnosis and so on. You don't, yeah, it has to be efficient, and you want to label the slides right away. You don't want to work with stickers and these things where maybe the sticker is going to come off, but here you can directly write on here. Yeah, so that is the point of this. Yeah, and they sent me a whole different ones. Let me just a second. Here's another one. Yeah, and yes, these seem to be non-frosted. They have an orange one, and they seem to be, they seem to be semi-transparent here. Yeah. So yeah, sometimes it's basically there's no paint on here, but sometimes they also simply made it rough, the glass, so that you can also write on it with pencil. Yeah. Or pen. Yeah. So, as a matter of fact, pencil is pretty good for labeling those, because pencil is not solvent soluble. So, if you accidentally spill water over it or alcohol, the pencil will not come off. Yeah. Let me quickly have a look here. So, that's you will buy both surfaces. I talked about the biofilm already on both surfaces. The cardboard version of this, of this rack, as well. Do they come back to life? I don't know, and I think I'm going to try this out. I'm going to take some of the chlorella algae and put it again into one of those containers here, okay? And then we're going to see what they do. Directly into the pond, because it's not practical. You could do that, I guess. Yeah, that you try to collect the biofilm in a natural environment. But then again, you might have snails there, and those snails, they actually eat off the biofilm again. Yeah. So tape the surface. I'm going to skip over those questions that I already answered. Yeah. Fold cardboard into a V and cut out circles along the crease. Stand it upside down. Ah, that's a nice idea. Didn't think of that. You know what? I'm gonna. Do I have somewhere? Look what I got here. I get the idea. I'm gonna illustrate this. Thank you very much. Look, these are my tardigrades here. The idea is the following. Where's my scissor? This is a nice one. You know what? I'm gonna just. So this is a how do you say, improvised version. So what you do is the following. Something like this. I've not done this before yet, but this is actually nice, and never thought about this. Something like this maybe. And it might be a little too small. So, and I mean, this is the cardboard is too small, obviously. Nah, of course not. This is not gonna work. Yeah, but you get the idea. This is way too small. But for illustration purposes, you make it larger, obviously, and then you cut this in, and then you can essentially try to, put your tubes in here. Yeah, and if it goes much higher, then of course it's going to hold the tube. Yeah, this was a little bit the idea. Yeah, this is actually quite a nice idea. I like that. I didn't think of that. Yeah, yeah. Maybe something like this. I don't know. You get the idea. Who cares? Yeah, you get the idea. Where was this again? That's here. You can put the thin silicone. Yes, leave it on the pond a lot. Put one slide on an onion mesh bag and a string. Leave in. I like to leave it in the pond idea. One side in the onion mesh bag and a string, leave it in a month, or sponge and string. So basically you submerge the whole thing. The thing why I like to think about growing biofilm directly on a slide is because you kind of keep the natural, how do you call this, the natural arrangement of the cells. Yeah. Okay, could use a latex-based paint then. Okay. What's better, to use the filter paper or agar? I talked about that. It really both works. I would use the filter paper because it's simply easier to do. I used coffee filter. So yes, I don't know why the screen with the slime mold was a little bluish. I think that's because maybe it was some kind of a white balance issue. Yeah. Silicone you would use to fix the slide inside the fishbowl. Okay, so that's basically for making the biofilm here. Video to show how microscopes work. I'm not just an expert with the world's depth of field. You know what I could do is, by the way, for those of you who don't know, I have a second channel called Microbehunter Microscopy, which is a slightly more technical microscopy channel. I usually answer questions there, and that's something that I might want to pick up there as well. So I'm also doing microscope reviews, videos in the other channel, and I talk a little bit about the optics as well. So these things here will probably not go into this channel here, but into the other one. Yeah. Did you experience any difficulty growing water fleas or Daphnia? They are such a fuss, they easily die. Yes, I do confirm. I just show you something. Yeah, and here, yeah, I don't know if you're able to see them. Maybe you're able to see them. Yeah, but that's basic. Let me see if can I turn on the light here. I don't know. Yeah, you might be able to see. Let me see. Let's put this down here. Yeah, so that's basically these are Daphnia here, and yeah, basically what I do is the only thing that I do is I add a little bit of dry yeast every couple of days. Yeah. And don't overfeed them. That's really important. If you, as soon as you start overfeeding them, yeah, they're gonna die off. Yeah, because by overfeeding, you're growing a lot too many bacteria, and then the bacteria they take away too much of the oxygen. Yeah? So you, if you see that the population goes down, then you add a little bit of yeast again. Yeah, and then you kind of try to push the population up again. Yeah, and sometimes what I also do is, and I made recently a video on this as well, is I add sometimes some of these, let me move this up again, yeah, some of these, scratch off some of these chlorella algae, yeah, and this is how I was able to maintain now this culture here maybe for a couple of months, yeah, but it's a little, I admit it's a little bit delicate, yeah, yeah, so. That's why I've got, I think, 3 jars now over here with water fleas. The reason why I'm keeping those water fleas is not only just so that I have something to look at, but you might already know that I'm a biology teacher. And we also need them for biology lessons as well. Sometimes some of my biology colleagues also basically request some of the samples and specimens. So you see, that's actually also one of the reasons why I keep them. And if they need something, I take a few of them here to school, and then they have something to look at with their students. Yeah. So that's a little bit so that you also understand a little bit of why I'm doing this here. It's not only for the live stream and for my own fun, but being a biology teacher, I have this double use, so to say, of the equipment that I have here. Yeah. So microscopes work. Did you experience any difficulty growing water fleas? If you acquire an old enlarger or manual camera lens, take out the glass, leaving the iris so the variable. Ah, yes. Yeah, that's another thing. Yeah. So basically, there is this, yeah, the aperture variable aperture diaphragm. Have you ever kept fresh water fairy shrimp? No, not shrimp. Over 20 years ago, I did keep my freshwater aquarium with fish. Okay. Yeah. So what you can try as well is, if you wanna, I've also tried that last year or so. I made a video there. Those called the brine shrimp, Artemia salina. They're called. They used to be sold under the trade name on the brand name sea monkeys. And they basically are also sold in the form of eggs with salt, and you basically, it's a ready-made mixture, and it also contains some food, because some people who have an aquarium, they will actually grow them as fish food. You can also do that, yeah. So yeah, and they're also quite nice to observe. I find myself feeling a little bad after looking at the wonderful, yeah, and then you kill them with 70% alcohol. Some of you might, yeah, why not answer that? Why is 70% alcohol used for disinfecting and not higher concentrations, like for example 96%? The reason is they found out that 70% is actually more efficient in killing microorganisms than 96%. The reason is because 70% alcohol corresponds to the solubility of the cell membrane. So alcohol is able to dissolve the cell membrane, and if you use 70% alcohol, then the membrane of the cell is most soluble, and therefore it has the highest efficiency in disinfecting. Yeah, because what you're doing is by adding the alcohol, you're stripping the cells of the cell membrane. Yeah. Ah, yes, I forgot to label the things. Yeah, that's yeah. Hydra, are quite nice. I got a few years ago some Hydra from a colleague at work who basically had them from his home aquarium. Yeah. So, and I put them under the microscope as well. And the Hydra, I also got them sometimes in ponds. And what you have to do is you have to collect some dead wood or some leaves or so, some solid material from a pond. And if you're lucky, you're able to find Hydra growing on there because it likes to attach to the surface. Yeah? They're quite large. Well, the individual cells are fairly small. Yeah, that's quite cool to see. Okay, the microplastics crisis can be fixed. I don't know. Look, the whole thing with the microplastics, I've made already a few videos some time ago. You won't believe you find them everywhere. You just have to make sure that you're not if the slide is completely clean, okay, and you put a water sample on here, you're likely to find some of this somewhere, right? And as a matter of fact, I also talked to a co-worker, and I was told that at university she's basically, when you put sea salt, basically marine salt, which you can buy in shops, under the microscope, apparently you can find also quite a bit of microplastics in there as well, unfortunately. Yeah. So I don't know if it can be solved or not, but especially clothing, the fibers from textile fibers like polyester and those synthetic fibers, that's a large percentage of the microplastics as well. Yeah. So again, here we're back at the sample, and I'm going to now do the following because I've been talking over 1.5 hours again. Yeah. There's been a request. Let's continue with the samples. Okay. So you know what I'm going to do? Alcohol to the water fleas to slow the heart down. Yes, you can add a little bit of, I've also done this. You can add alcohol to some water fleas to slow the heart down. And you can add caffeine to the water fleas to speed the heartbeat up. Yeah, that's a common experiment. So there has been the request for me, again, to put something under the microscope here. I don't know which one I did or which one I did not. Maybe the last sample today, I don't know, was it this one here? And what I'm going to try now is I'm going to not try to take I'm going to try the following. So this one here, is also quite, the more ugly looking, the better. You can't see it. Yeah, but I don't know if this is enough really. Yeah, so let's take. Look at this. This is ugly. I love it. This is the most improvised version. Normally, if you do this, sometimes you're able to wash out some of the. I don't see a lot here. This was not a good one. You see, here is the tissue paper. Yeah. So the white background will also tell you a little bit if we've got a sample on here or not. So let's do it like this. Okay. Usually on the solid material, maybe you are able to find some organisms because sometimes they like to grow on a surface. Like this, which one was it? Was this one or this one? No, that was this one here. So, and I need a cover glass. Here are the small ones. Yeah, some of you already have to leave. I wish you in those have to depart. I wish you in any case, if you're already leaving, a very nice week. And I'm going to stop as well in a minute or so. But yeah, there has been the request for me to put something, and I have no idea what to expect. I'll be honest with you. No idea what to expect. That's one of these nice surprises usually. Way too much water on here. And yeah, maybe there won't be anything interesting here. But maybe there will. I don't know. This is mostly... Let's go down with the magnification. So like this. Like this. Ah, yeah, interesting as well. Ah, there is some movement going. Some worms. Yeah, that's usually those worms. They are decomposers. What are these? What are these lines here? Are these? Could these be also some kind of? They also look like cyanobacteria. Oh, sure they are. Yeah. Sure enough. A little bit too much water. Therefore, some of them are a little bit out of focus. Yep. It's a lot of them. And then this would be again the time to make a little, way too thick of a water sample. Some rotifers here as well. Another rotifer. What's going on here? Nematode worm. Yeah. So those guys survived, yeah, I would say several months in this container. Here another one. Yeah. This is again the possibility to do a time-lapse video to see if these cyanobacteria, as I suppose them to be, simply from the size, then maybe they're also gliding around. Yeah, yeah. Okay, I think yeah, that's basically here in this sample lots of rotifers. Okay, so and I think I'm going to now do the following. Let me quickly see if I forgot something. Here, Marie is asking, what would you suggest to part as a good microscope activity for biology class, unless I missed your reply in any way? You know what? So many of them. Which good microscope activity for... It depends if it's a beginner's or not a beginner's class, and usually what I do at the very beginning is I allow them to simply look at ready-made microscope slides, my students, simply so that they can do some, not sample preparation, but simply observe some things. And then usually what I do is the students should look at their own hair under the microscope. Yeah, so they should take a little bit of their hair, put it under the microscope, and then what I do is allow them, there is, especially if the student is a little bit older, there is a way how you can actually calculate the diameter of your hair by using a ruler as a reference. So you take a picture of a ruler under the microscope, you put the ruler directly under the microscope, and you take a picture of your hair under the microscope, and you do a little bit of direct proportion calculation, and then you can determine the diameter of your hair. So usually I'm doing activities like this. Yeah. So what I'm going to do, people, I would like to, I hope I answered all of the questions here. I would like to, in any case, wish you all the best. I think I'm going to call it a day, quits for the day. It's already quite late where I am. I wish you a nice week. In any case, more videos are coming on this channel, regular videos. But for today, I would like to wish you all the best. Okay, and all the best and happy microbe hunting as always, and hopefully to see you again next week. Bye bye.